तुमच्यावर आता तुम्ही हे तुमचं इक्वल हॅलो हॅलो
आता बोल आता बघा नाही आता तुम्ही बोलणार
सरळ आहे पुढं
I, Dr. and please welcome all for this conference. Relevance of AI and Biotech in Agriculture and Medicine. REBIOAM-2026 is a two-day conference organized by the Department of Biotechnology, VGS-National College of Arts, Commerce and Science, in association with we begin today's the first session being delivered by Dr. Bani Jolly. And please to introduce you to Dr. Bani. Dr. Bani is the second head of this research and development, Genome and senior at Karkinos, Sandhya R. She is passionate about developing informatics and AI solutions for supporting the democratization of whole genome sequencing for all. Her contributions to scientific literature encompass diverse areas ranging from genome characterization to genetic epidemiology. Apart from the detection and characterization of emerging SARS-CoV lineages in India and contributing to the international surveillance and technical efforts as part of the WHO programs in Southeast Asia. She has also been a part of large national and international efforts for genetic epidemiology, including human and pathogen genomes. I will at this and I will to begin. Thank you.
Thank you so much for that very kind introduction, and I'd like to thank you all for having me and giving me the opportunity to present this work here today. So, good morning, everyone, and we are actually now living in a very interesting time to grow up in biology, because for the first time we have realized that biology is no longer something that we observe, but it's something we can read, analyze, and increasingly understand. And when I say growing up in the era of intelligent biology, I just don't mean as children or newborns. I mean all of us, researchers, scientists, technologists, students, clinicians, all growing into a world where biology itself has become so data rich and computational. And basically, genomics has given us the code of life, and artificial intelligence is what is finally helping us make sense of it. So today I'll walk you through how genomics and artificial intelligence are coming together, not just as buzzwords that we hear around every day, but as necessities to address problems that biology created at a scale humans were never meant to handle alone. So before we talk about the present and the future, let's go a bit back to 1963. So this is Dr. Robert Guthrie, and what he introduced looks almost very trivial today. What he invented was a small heel prick test in which he took a few drops of blood on a filter paper to screen for a very serious condition in newborns. This was a very radical idea for its time, that what if we could detect the disease even before a child ever showed symptoms. So Dr. Roberts' test for phenylketonuria proved something very powerful, that early biological insight can completely change a life's trajectory. This wasn't genomics yet, it was not artificial intelligence yet, but it was the first time medicine actually said that don't wait for disease to declare itself, go looking for it early. And just a decade earlier, in 1952, this image was taken. And what we famously know Rosalind Franklin. This is her photo 51, which was the first time humanity was able to see the structure of DNA and the physical basis of heredity itself. So this was a different kind of breakthrough. Dr. Robert showed us when to intervene, and Dr. Franklin showed us what exactly were we intervening in. So together, these moments quietly set the stage for everything that followed: genomics, sequencing, whole genomes, and so on. And eventually, they led us into a new problem. But far before that also, by the 1930s, another quiet revolution was also underway. Not in biology, but this guy called Alan Turing was trying not to solve medical problems. He was just trying to ask a much more fundamental question about life. That can machines think? And the machines he built were designed to actually break codes, patterns that are far too complex and far too fast for humans to decode reliably. And that idea turns out to matter now a lot for biology, because once we learn to read the DNA, thanks to Franklin and all the subsequent work that followed in genomics, we decided to look early thanks to Dr. Robert, and now we've created a new problem. So biology itself has become a code, and that too a very massive one. And just like wartime encryption, decryption that used to go on, it quickly crossed a scale that human reasoning alone is no longer enough. So Turing didn't build AI for genomics or biology, but he gave us the blueprint of what would eventually make biological data or genomic data very, very usable. And by 2010, genomics has quietly stepped out of research labs into people's actual lives. So when this actress called Glenn Close publicly spoke about undergoing a whole genome sequencing test, something very important happened. So health stopped being a very abstract concept that is only reserved for patients, and now it became deeply personal. For the first time, people weren't just learning about disease, they were learning about the probability of a disease happening and the uncertainty of it written into their DNA. And they realized that healthcare and genomics is not just for patients, but it can be actually for everyone. So when we look back, it's tempting to see all these moments in history as isolated breakthroughs. But what this timeline really shows is something else completely. Progress in biology was never driven by one discipline or one kind of mind. So, for example, a mathematician taught machines to actually think. A scientist showed us the structure of DNA. A microbiologist proved that early screening could change and save lives. And decades later, a public figure reminded us that genomics isn't an abstract concept, it is a very personal thing. And none of these people were trying to build intelligent biology, but together the work they did eventually helped do that. So what we're standing on today is the combined work of not just biology, but also computation, medicine, and societal changes, slowly moving us towards care that is proactive, preventive, and very, very personal. And as we reach this point on the timeline, one thing becomes very clear, that the future of healthcare won't be built by just biology alone. It will be built by many minds working together on the same problem with intelligence woven into the system itself. Now, before I go on to the solutions, let's first talk about the problems that we need to solve very urgently. So I'll ask a very rhetoric question, that if you could go back in time to change just one moment in a baby's healthcare journey, would you do that? And most of us, I know, would not hesitate to say yes, we would. So let's take it by an example. So for example, a baby is born. They look very healthy, they cry, they feed. Standard newborn screening tests are done, which is your basic blood tests, and everything comes back as very, very normal. The parents are reassured, as this should be. But over the next few weeks, very small things start to appear. The baby is not feeding very easily. Weight gain is very slow. There is crying more than expected. It's crying more than expected. And most people brush it off as acid reflux, something that babies really have in general. But by 3 to 5 months since birth, the picture starts becoming a little harder to ignore. The baby is not achieving the milestones it should typically do in this time frame. The muscle tone seems low, baby is not making eye contact, but still the parents are told that every baby develops very, very differently. So it might be that the baby is just normally developing. But at 6 to 8 months, the red flags are now becoming much, much clearer. The baby is starting to have staring spells. There are developmental concerns, but there are still no clear answers. Why is this even happening? Tests are ordered, but scans show very non-specific changes. And this is the hardest part, that no one did anything wrong till here. But as the baby grows, the symptoms escalate. Clarity does not come. Around 9 to 12 months later since birth, seizures begin. More tests are ordered, metabolic panels are done, imaging is done, nothing very definitive comes out of it. The family has now started to move between specialists, neurology, genetics, therapy, searching for an answer that still does not have a name even. And it's only much later, often 18 months or more, that a genetic cause is seriously suspected, usually after referral to a tertiary center. And even after that, the question is that what is the diagnosis for this child? And it's not even the most important question. The most important question is, when is this diagnosis even reached? Does it take 2 years, 3 years, 5 years, even longer, or even never in some cases or some countries? But by this point, the most critical windows for brain development have already closed for this child, because interventions will now begin late and some developmental loss is very, very irreversible. So the cost here was not just medical, it was emotional, financial, and lifelong for this family. This is what a delayed diagnosis really looks like. Not a single missed test, but a system that finds answers too late because it was never designed to look early enough. So let's rewind the story. The same baby is born. He is healthy, active, reassuringly normal on a standard newborn screening test, just like we saw in the previous slides. But this time, the parents are offered an option that would, along with routine screening checks, do you also want to go in for a genomic newborn screening? And the couple does agree to it. Within the first couple of weeks, the genomic insights arrive, and the results show that the child carries a pathogenic or a disease-causing variant in a gene called CDKL5. And long before symptoms could even appear, this particular mutation has already been caught in this child. So with early genomic diagnosis, the timeline for this child has now completely changed. By week 3, the family is already connected to a pediatric neurologist who specializes in disorders related to this particular gene. So this is not after months of referrals, but immediately that they are now able to connect to a very particular doctor. So by week 4, treatment begins before the seizures even start in this child, because the parents know what to watch out for. They are prepared. They don't have any surprises left to wait for. By the 5th week, a targeted anti-epileptic approach is initiated by the doctor, and the family has access to very experimental therapies and clinical trials that would otherwise come much, much later if the diagnosis was reached at all. So at 1 year, the diagnosis hasn't really disappeared. All the challenges are still there, but there is also something that did not exist in the earlier story, which is a care plan, a pathway, and a team that is acting ahead of the disease and not really chasing to understand what it is. And this is what early genomic insights really offers. Not perfection, but preparedness. So what's important to notice here is that the same child, the same genetics, the same condition, the only thing we know that changes is when we know what disease the person or the child is going to have. So in the first timeline, without the genomic screening, the diagnosis comes too late, and this is when critical neurodevelopmental windows have already closed. The interventions or management strategies start after the damage has occurred. The cost accumulates not just medically but also emotionally. In the second timeline, we have seen that with genomic screening, those early windows are completely protected because care begins before the symptoms even arise. Developmental gains are maximized and complications are reduced. And families are supported early instead of being overwhelmed later of having to run around looking for answers. So this is the real impact of intelligent biology. Not changing how our biology looks like, but changing the trajectory of life of a person by changing the timing of understanding what can happen in the future. And once you see this difference, it is very hard to argue that while we grow up in the era of fast evolving tech, early genomic insight should be optional. And this is not just a hypothetical story. This is baby Margot. She was born in August 2023 and she was diagnosed with CDKL5 deficiency disorder. Because she was offered early genomic insights and coordinated care, Margot's journey looks very different from the one we saw earlier. So today she is more than 2 years old, I think. She is very curious, she's expressive, she's chatty, and she loves to struggle with her parents. And all this happened because the diagnosis did not really disappear, but the challenges did not end. But what we changed was the trajectory of her life. Earlier understanding, earlier care, and a family that wasn't left navigating uncertainty alone. So this is what intelligent biology looks like in the real world. Not just abstract innovation, but real children, real families, and real futures being shaped just by the factor of timing. So this is what I meant by my this is the point I want to make by this story here, that with the tools that we have today, the future may not need fixing because we start getting things right earlier. So genomics, for example, lets us see this risk before it becomes a reality. And artificial intelligence now helps us understand that information at a scale that humans can never do alone. So when we combine the two very thoughtfully, we stop chasing disease and start shaping healthier beginnings for everyone. And that's what growing up in the era of intelligent biology really, really means. So before I delve into the next problem, what exactly is genomics? It is at the core of everything we've talked about, and which is the human genome. So every one of us carries the same basic blueprint, which is 23 pairs of chromosomes made up of genes written in about 3.3 billion base pairs of ATGCs. So this code influences things that we think of as very, very simple also, like eye color, hair color, why we look like our parents, and so on. But it also shapes things that matter much, much more, which is how our bodies develop, how we respond to disease, and how we respond to treatment. So what is important to remember is that genome itself isn't rare or special, but variations in this particular genome is. And each person carries millions of differences or variations in this DNA code. Most are harmless, some are protective, and a fraction of them can profoundly affect health. And the challenge isn't reading the genome anymore. We've already solved that. Traditionally, genetic testing has been done in layers. You start with a small set of genes, look at only a fraction of the genome. If that is negative, people generally have moved on to something called as exome sequencing, which is able to cover 1 to 2% of your DNA only. And now this technology called whole genome sequencing changes this concept entirely. Because in a single test now, we are able to capture almost all of your genome, not just the genes, but the regions around them, including structural changes, things that repeat for a very long time, deletions, insertions, and even the mitochondrial DNA, which influences how your body processes energy levels. Now, this matters because disease does not really restrict itself to neat categories of a particular set of genes. Some conditions are driven by single base changes, others by large structural variations, and so on. So whole genome sequencing gives us one comprehensive view instead of multiple fragmented tests over time. And here is the catch now, that the power of whole genome sequencing isn't just in generating data, it is actually in interpreting it. So this is a simple way to think about what variations in the genome really mean. So imagine a word, the genome like a word, and changing one letter or making a substitution might change a word from cake to coke or joke. The word changes, but the meaning is still pretty much very clear. But there are other changes that are much more disruptive. A deletion or an insertion might change the word we started from cake to pick into sick. And now that the meaning is very, very different, the difference can actually matter a lot biologically. And this is exactly what happens in our DNA. Most variants are harmless. Some of them are sitting in the gray zone where we don't really know if they're harmless or not. And a small fraction of them are truly disease-causing or pathogenic. So the challenge is that when we sequence a whole genome, we don't just get one change, we get millions of them. And deciding which changes are just benign noise, which of them might cause disease, is something that we now need to do at scale. Because this is the basis of understanding how genomics can help our understanding of particular diseases, especially genetic ones. And why this is a problem is that we often talk about rare diseases as if they are a very niche problem. But when you step back and look at the full picture of it, the numbers tell a very different story. Collectively, genetic and rare diseases affect close to 400 million people worldwide. In India alone, that number is estimated to be 70 million. This number is so huge that in this perspective of the world, this is, if all of them were to live in a single country, this country would have a population of more than USA. And if all the rare disease patients in India were to live in one state, it would be more than the population of even Karnataka. So this is not rare. This is a public health crisis hiding in plain sight. And what makes all of this even harder is the journey that these patients have to go through. So on an average, a person with a rare disease sees multiple doctors, receives several misdiagnoses, and waits years before reaching the right answer. And during that time, symptoms progress, families lose trust, and a critical treatment window is often missed, especially in children. So the problem isn't a lack of care or effort. It is just that rare diseases are genetically complex, individually uncommon, and incredibly difficult to recognize without doing genomics, which is why solving rare diseases at scale was never going to be possible with traditional approaches alone. It requires intelligence that can connect patterns across millions of patients and genomes. And this is a reminder of why all of this matters, because on an average, it takes 5 to 7 years for a patient with a rare disease to even reach a diagnosis. That's why most of a childhood is spent without answers. Nearly 30% of children with rare diseases don't survive past their 5th birthday, not because their conditions are untreatable, but because they are not identified at all. And if we speak in the neonatal ICU units, about 1 in 3 critically ill infants with a suspected genetic disorder will die before a diagnosis is even made. These aren't edge cases. These are failures of timing and interpretation and lack of using a technology that can actually change the trajectory for these patients. And these are exactly the kind of problems that intelligent biology, genomics, and AI are meant to solve, not someday in the future, but now. And one common misconception about genomics and healthcare is that it is useful only when someone is unwell or is a patient, for example. But genomes aren't just about diagnosing disease, they're also about understanding your risk before it becomes an illness. So, for example, in our institute itself, we had a healthy, active 50-year-old who came in for a whole genome sequencing test. They had absolutely no symptoms all their life, no red flags, and in fact, they wanted to get even more fitter when it came to their physical exercises. So, genomics is something that was able to reveal inherited risks in a person. So, for example, it was revealed that this person has a mutation that causes a condition called hypertrophic cardiomyopathy. This is a thing that would not have shown up on a routine health checkup. And this is a condition in which your heart muscles grow thicker than they should normally do, which puts a lot of strain on your heart, meaning that if in case you do very rigorous physical exercises, your heart might just give out. So the value here isn't fear or over testing, it is now foresight. Knowing this early allows for very simple, sensible steps that the person can now take, including regular monitoring, lifestyle adjustments, more importantly, not running marathons, definitely, and starting preventive care long before anything goes wrong. So this genomic insight isn't just about labeling people as sick. It is about keeping healthy people healthy for longer and avoiding any adverse events from happening in the future. So that's why genomics isn't just for patients, it is for everyone. And conditions like cardiomyopathy are very important to screen through genomics because we read about it every day in newspapers, young people collapsing after doing rigorous exercise, dying of sudden cardiac death, and we think of them as isolated events, but they are actually very, very relevant in the population when we think about it genomically. And this is because one of the most important shifts in genomics is realizing that actionable findings are not rare. They are very common, especially in Indians. So roughly 1 in 20 people carry a genetic finding that can meaningfully change medical care. This includes conditions like cardiomyopathy, inherited cancer risk, other cardiovascular conditions, treatable metabolic or neurological conditions, and even how we respond to certain commonly used medications. All of these individuals don't really feel unwell and they wouldn't be identified through routine checkups. But with genomics, these risks become visible very, very early. So when monitoring, prevention, or simple interventions can actually make a difference. So in that sense, genome medicine isn't just a niche area of healthcare, it's modern healthcare in practice, I'll say. And this is what genomics has enabled us to do when it is used well. Most diseases follow a very predictable path from health to early changes to symptoms and then finally, eventually, chronic illness. Traditionally, we intervene very far late in this journey that nothing can be done. But genomics shifts that timeline upstream. Your DNA holds clues about your future health risk long before the symptoms even appear, and when the opportunity to preserve health is the greatest. This is, sorry, this is what we call as the golden window, where small informed actions can prevent or delay disease entirely. And that is when the best time to get your genome sequenced really was yesterday. But the next best time to get this genomic insight is when we finally have the tools to interpret and all of that information intelligently and then act upon it very responsibly. So the next best time is basically today. This is healthcare that predicts, prepares, and personalizes, not just after the disease appears, but before it even has started to show subtle signs or symptoms. But when we now step back and look at the full picture, one thing becomes very, very clear, that genomics, healthcare, all of this was never a human-sized problem. Each genome carries millions of variants. Rare diseases don't often look clinically alike, but they are they might look clinically alike, but they can be completely different genetically. Scientific knowledge keeps evolving faster than any individual can track or is humanly possible to track. And most variants we encounter are of uncertain significance. Either they are novel or they are very, very context dependent. So to make sense of any one genome, we need population scale data, constant reevaluation, and the ability to integrate signals across biology, medicine, and time. And no matter how skilled, dedicated, or persistent we are, humans alone are never going to do this reliably, consistently, and more importantly, at scale. And the advantage of having all these tools and technologies is only if we can do it for everyone in the world and not just for the elite or the people who can afford to do so. So the challenge here isn't a lack of expertise, it is the size of the problem itself. And this is the moment where everything now has started to come together, when biology, or human biology, in fact, has started to meet with artificial intelligence. So AI just didn't arrive to replace clinicians or geneticists or scientists. It arrived because biology reached a scale where learning had to become very, very continuous. So intelligent systems can learn from every genome analyzed, integrate genetic data with clinical features, population context, and adapt as scientific knowledge evolves, something humans can't possibly do. Most importantly, they allow interpretation to scale without scaling uncertainty. So this is not automation for the sake of speed. It is basically augmentation, giving human expertise the support it needs to work at a biological scale. And this is what intelligent biology means. So one of the most powerful shifts enabled by AI and genomics is moving healthcare from reaction to prediction. Most genetic and chronic diseases begin years before symptoms even appear. So traditionally, medicine steps in only after the damage has already been done. But by integrating genomic risk with clinical and population data, AI models can allow us to identify disease susceptibility even before the symptoms start to emerge. And that changes the goal of care from treating illness to maintaining wellness. So it is not about predicting the future with certainty, it is about shifting decisions upstream. And this is what predictive health looks like in practice. And the second problem that AI can now solve is that rare diseases are called rare, but the journeys are long and painfully very common. Millions of patients spend years moving between clinics, carrying uncertainty instead of answers. And AI can absolutely change this by connecting pieces of information together that humans can't easily hold together in their brains. So by structuring clinical notes, for example, mapping symptoms to standard phenotype terms and matching them against the genomic variants that have been uncovered by in a person's genome, and integrating rapidly growing global data sets with it, AI can now very quickly narrow down the most relevant genes and variations for a particular patient. So what once took years of sequential answers and testing and referrals can be compressed into weeks and sometimes days. And this does not replace clinical expertise. It focuses it on something that needs to be looked at very, very importantly. Now, over the last 2 decades, we have solved one very hard problem that has existed in the previous century. That is how to read the genome quickly and affordably, that it can be done for everyone. So the cost of sequencing has now collapsed. Whole genome sequencing has actually become routine, even though people have not really realized it. In India itself, what costed 1.5 lakhs for a genome till some months ago, it can today be done for a mere 20,000 rupees. But at the same time, another curve has shot up here. The number of genomes being done and the number of variations we can now uncover in every individual. So a single whole genome test reveals millions of variations in a person. Most of them are rare, many of them are uncertain, and only a small fraction of them are immediately interpretable. So this is the problem of genomic interpretation now. Reading the genome is now very, very easy and affordable, but understanding what actually matters for a patient is still not very easy. And this is the point where biology clearly outpaced human cognition, not because we lacked expertise, but because we did not realize the scale that we will have to work on. And now this is what intelligence at genomic scale can and will look like. When a whole genome produces millions of variants, the goal isn't to look at everything. It is to surface what matters. And AI systems do this by filtering, ranking, contextualizing all of this genomic information using phenotype or symptomatic data, population data, and existing biological knowledge. So in other words, they separate signal from noise. And tools like AI Marvel are designed specifically for this, for for creating a priority list of candidate genes, especially in Mendelian disease, by combining this genomic data with clinical features in a way that is not feasible to do manually. So the result isn't automation of all of this for its own sake, but it's simply focus, allowing clinicians, geneticists, other doctors to spend their time on the variants that are most likely to explain a person's phenotype. And this is how interpretation now becomes scalable without losing accuracy and rigor. And the next thing that AI can solve is that newborns often look very healthy, and most of them generally are. But some of the most serious genetic conditions are completely invisible at birth, and by the time the symptoms appear, critical windows for development and intervention have already closed, like we saw in the example that I gave in my earlier slides. And this is where AI-powered newborn genomics can change the equation completely. By analyzing genomic data early, AI can prioritize actionable variants, identify carrier risk, assess all types of disease risk, and surface insights that matter the most during the treatable window. So the goal isn't to medicalize infancy, it is to make sure that when action is possible, we don't miss the moment. And this is preventive care occurring from day one, not reacting to the disease, but protecting developments ever since we are born. And one very important shift that is happening right now, even though we might not see it very clearly, that cancer genomics is no longer dominated by Western data sets alone, which is something that has been historically true. In India, initiatives like the Bharat Cancer Genome Atlas are beginning to capture the unique genomic landscapes of cancer seen in our population. This matters because cancer biology is not identical across geographies. Variants, mutational patterns, treatment responses can differ, and without representative data, precision medicine remains incomplete, especially for Indians. And as Indian cancer genomic data grows, it creates opportunity and also responsibility. The opportunity is to develop diagnostics and therapies that are truly relevant to Indian patients. And the challenge is that this data is growing at a scale that is so unprecedented that we need intelligent systems to start interpreting all of this data. So this data is incredibly valuable, but alone it does not improve outcomes. We need interpretation. And as these data sets grow, so does the complexity of all of it. Thousands of mutations per tumor, population-specific patterns, and evolving resistance mechanisms is all that needs to be captured. And this is exactly where AI-driven precision oncology becomes very, very essential. Because AI now allows us to learn from Indian cancer data at scale, identify driver mutations, identify therapy targets that are very population specific, resistance patterns that are relevant to our patients, and not just those represented in global data sets. So in other words, population scale data only becomes precision medicine when it is paired with population scale intelligence. And one of the most common and the most frustrating problem in genomics, especially while doing whole genomes, is the outcomes of this genetic testing, which is something called as a variant of uncertain significance. So to put it simply, this is a genetic change or a mutation or a variant where we don't yet have enough evidence to say whether it is harmless or disease causing. It looks a bit like looking at a random stray cat and asking, is it friendly or is it aggressive? Without context, without really getting in contact with the cat, you can't really tell if it is harmless or if it is harmful. And guessing will be very, very irresponsible to do so. So around 20% of genetic tests identify only these uncertain variants, especially as we move to large panels like whole genome sequencing. And that uncertainty matters because it creates anxiety from families, hesitation for clinicians, and a heavy burden on genetic counselors who have to explain that what we don't know about this variant as carefully as what we do. So this isn't a failure of genomics, it's a consequence of doing genomics at scale. When you look broadly, you inevitably find things that science hasn't fully interpreted yet. So the real challenge is how we move from variants of uncertainty to actually creating systems that can interpret these variants very, very carefully moving forward. And so variants of uncertain significance aren't really a dead end. They are something we can think of as snapshot of what science knows today. It is something that is just limited by current knowledge and can be improved in the future with as and when knowledge and scientific evidence basically grows. So what we are learning now is that not all VUSs are equal. When you stratify them by evidence or symptomatic matches or gene relevance or population data, they start to behave very differently over time. And this is where AI becomes very, very powerful, but because by structuring clinical notes, mapping symptoms to standardized phenotype terms, and integrating all of this with the genomic data, AI can now grade variants of uncertain significance as more or less likely to be clinically relevant. So instead of a single bucket of uncertainty, we can move from strong and weak categories, which can directly inform follow-up, surveillance, and reanalysis. Importantly, this approach is grounded in evidence, and recent work shows that VUS subclassification improves reclassification rates and clinical utility over time. So the goal isn't to force certainty where it does not exist. It is to manage uncertainty intelligently, transparently, and doing so at scale. And one of the hardest problems in genomics is that many variants simply don't have any gene-disease relationships yet, because human interpretation is limited to what has already been described in literature. But AI isn't. AI is very, very good at looking at patterns that humans cannot see. So by learning from large-scale genomic and clinical data sets, AI can now uncover new gene and symptom relationships or associations and patterns that have not been explicitly defined before. So in studies like this one, for example, that we did, deep learning models can analyze genomic variations at scale, identify relevant genes, and most importantly, use interpretability methods to explain why these new genes matter in a particular disease. So this isn't about black box predictions, it is about accelerating discovery or turning weak signals into hypotheses and hypotheses into new biological knowledge. So in that sense, AI doesn't just interpret genomes, it expands what we already know about human biology itself. And one of the most powerful ideas in modern genomics is that this isn't just a test that has a one-time value. It is a lifelong thing that you have. So from birth to old age, the same genome can inform very different decisions at different points in life. Early on, it can help identify very serious genetic conditions. In childhood or adulthood, it can inform risk for common diseases and guides prevention and lifestyle choices. Later on, the same data can support decisions around carrier screening, cancer risk, transplantation, and how we respond to medications, and a lot of other things. So what changes over time isn't our DNA, it is our understanding of it. And this is where AI becomes very, very essential. And why do I say so is that one of the most fundamental mismatches in genomics today is this. Scientific knowledge evolves very, very continuously, but the patient reports are often frozen in time. So reanalysis of genomic data is a very big problem. A variant that is uncertain today might become clearly meaningful tomorrow as evidence grows, but only if we go back and look at that data again. And this is again where AI changes this model entirely. Instead of static reports, we move to living genomic intelligence, basically systems that can continuously reinterpret genomic data as new evidence starts to emerge. As databases grow, studies are published, new associations are discovered, AI can reprocess existing genomes and surface new insights automatically. So the genome itself is not changing, but our understanding is completely evolving. And what is not humanly possible to do so when intelligence is built into that process, a genome becomes something that learns over time, delivering increasing value across a patient's life. And that is the difference between sequencing the DNA once and truly understanding it over a lifetime. So, I will summarize all of this by making you step back and look at the full journey. This is all it's really about, is moving from codes to clinics. Research is something that fuels discovery. It is where new knowledge begins. Genomics is something that allows the knowledge to become personal, tailored to each individual. And AI plays a very critical role here by turning massive amounts of biological data into something we can actually understand and act on. And none of this work is done in isolation. It's collaboration between doctors, scientists, engineers, patients, all of these people who bring these advances to life. So ultimately, the future of biology will not be built by machines alone, and it won't be built by humans working alone either. It will be built only by those who combine human judgment with the tools and techniques we have with machine intelligence to make biology work at a real world scale. I'll stop here and I'm happy to take any questions. Thank you.
Am I audible? Dr. Bani, am I audible to you?
Yes, sir, you're audible.
Uh, First of all, a very big thanks to you for such an informative explanation. As an academician point of view, I would like to ask one question. You mentioned that in modern days, the sequencing, whole genome sequencing has become very cost effective.
Right.
So for our students, we would like to know what exactly has advanced in terms of analysis point of view.
Okay, so basically, the companies that provide this genome sequencing technology have not just improved the biology and the chemistry of it, they have also improved the quality of the tools that are used to analyze all of this. And I'm talking from a computational perspective. Now that we have hardware that is very, very scalable to interpret things in a very short time, the thing that has changed is not just the speed at which we can do sequencing, but also the speed at which we can analyze and process and interpret these genomes. So, for example, in India itself, from sample collection to a report being sent out, the minimum time that has been shown is 36 hours. And that is a very critical time frame when we think about very urgent newborn cases, for example. So it's not just that the chemistry is constantly evolving. The technology that keeps evolving in this world is very much supporting all of this, not just by influencing scale, but eventually also influencing the costing of all of this per person.
Yes. Thanks. Thanks a lot.
Sure. Are there any more questions? I think I cannot hear everybody.
So if there are no further questions, I would like to thank Dr. Bani